I'm Dr. Zaheer Amjad, and today's topic is about pituitary gland and its functions, gross anatomy of the pituitary gland. You have already studied this topic, and this is just a revision of the topic. Our learning objectives of the topic are that at the end of the lecture, you should be able to know or give the location of the gland, where the gland is located, what are the relations of the gland, how do you divide the gland, and what are subdivisions of the gland, how the gland develops, briefly, what are the sources of development of the gland, what are the functions of the gland, and blood supply of the gland. So, hypophysis cerebri, or the pituitary gland, that is a small endocrine gland but of immense importance to the body, because this is the major gland of the body which controls all the other glands of the body, and it is present in close relation to the base of the brain, arising from the hypophysis, from the hypothalamus. As you can see here, this is the third ventricle, and from the floor of the third ventricle, you can see and from the hypothalamus, you can see the gland extending downwards. It is located in the hypophyseal or pituitary fossa, and it is called master endocrine orchestra because it is the master gland of the body which controls other glands in the body. All the homeostasis of the body depends upon the intact activity of the pituitary gland. The gland itself is controlled by the hypothalamic factors and hormones, and this is of vital importance to the body. Here you can see the location of the gland. This is sella turcica of the middle cranial fossa, and these are the anterior and posterior clinoid processes. In the center, there is a depression in the body of the sphenoid bone. That is called sella turcica. In a sagittal section, you can see it's like this, just above the sphenoid sinus. Below and in front is the sphenoid sinus, which is present in the body of the sphenoid. This is the sphenoid bone, and here is a depression which lodges the pituitary gland. So it is located in the middle cranial fossa, and that fossa is called sella turcica. As you can see here, this is the coronal section. This is again the body of the sphenoid, and this is the septum which is separating the two sides of the two paranasal sinuses, sphenoidal paranasal sinuses. These are the sinuses, air-filled sinuses, which open into the nose, and above these sinuses is the sella turcica, which lodges this pituitary gland. It is roofed by diaphragma celli. This roof is formed by the meningeal layer of the dura mater. There are two layers of the dura mater. One is that which is the endosteal layer, as you can see, which is just the periosteum of the gland. This is one layer which is always in contact with the bone. The other layer of the dura mater gets separated, and here below the pituitary, you see that there are connection between the two cavernous sinuses. These are the cavernous sinuses. On either side of this are the cavernous sinuses, which are spaces between the two layers of the dura mater on either side of the pituitary, below the pituitary, and at the roof of the pituitary, right? So this flange of tissue is the roof, which is called diaphragma celli. It is forming the roof of the cavernous sinus as well. And this is the wall, lateral wall. And here you can see below the pituitary, there is communication between the two cavernous sinuses These are the this is the intercavernous sinus. The other two layers of meninges, that is spire matter and arachnoid matter, they fuse together, and there is no subarachnoid space here, right? All the other two layers, they are fused together. And this is the cavernous sinus, which lodges the important structures over here. The internal carotid artery you can see here. We will go to the contents of this later on. Anyhow, it is roofed by the diaphragm ocelli, which is a layer of the dura mater, and stalk of the hypophysis cerebri pierces the diaphragm ocelli. This is the stalk of the, or the connection of the pituitary gland with the hypothalamus that is piercing this hole. There's a gap between the diaphragm ocelli, which is pierced by the stalk, and then it is lodged into the hypophysis fossa. It pierces the diaphragm ocelli and is attached above to the floor of the third ventricle, as you can see in another picture. It is oval in shape, as you can see, and 8 millimeter is the anteroposterior dimension, and 12 meter is transverse diameter of the pituitary gland, and weight is about 500 milligrams. So this is a small gland, but it has got a rich blood supply, which is a characteristic feature of any important endocrine gland. And if you come to the relations of the gland, then you see that superiorly, as I told you before, this is the diaphragm ocelli. Then above that diaphragm is the optic chiasma. These two optic nerves, they join to form a chiasma over here. So these are at the superior side of the pituitary gland. So any growth of the pituitary gland is definitely going to compress the pituitary, this chiasma and the optic nerves, right? Then there is tuber cinereum, the lower part of the hypothalamus, and infundibular recess. You can see here, this is the infundibular recess, right? And this is optic chiasma. And tuber cinereum is the name which is given to all this lower part of the, extending from the optic chiasma to the posterior region. That all is the tuber cinereum. And there is an infundibular recess, which is the extension of the third ventricle into the stalk of the pituitary gland. Then the subdivisions of the gland, on either side you can see the, on the either side you can see the cavernous sinus with all its contents, and below you can see is the sphenoid bone, body of the sphenoid bone, having the sphenoidal air sinuses, and even below that would be the cavity of the nose, or nasopharynx would be there below that. Excuse me. The gland has got two main parts, adenohypophysis and neurohypophysis. These two parts are according to the sites of, two different sites of origin of the gland. Adenohypophysis being outgrowth of the stomodium, ectoderm of the stomodium, and neurohypophysis is the downward extension of the diencephalon, right? The two parts, they are intimately connected together, but they have got different origins. One is coming from below, that is the adenohypophysis, anterior part, and the posterior part, which is the neurohypophysis, that is the downward extension of the diencephalon, right? So these are morphologically, embryologically, and functionally different parts of the gland. Their secretions are also different, their morphology is also different, and their embryological origin is also different. As you can see here, the two sites of origin, one is extending downwards, ectoderm, from the diencephalon is going downward, and Rathke's pouch, which is from the floor, roof of the stomodium, that is approaching upward, and then these two are incorporated together with a slight gap over there, and that constitutes the anterior, and this part, which is extending downwards, that constitutes the posterior part of the pituitary gland. So adenohypophysis and neurohypophysis are the two parts which develop from two different regions of the body. Adenohypophysis. Now this is the adenohypophysis anterior part, which is related to the optic chiasma. That consists of three different parts. They are morphologically and functionally, they're different. Origin is the same, but otherwise, the anterior lobe, the commonly called anterior lobe, or pars distalis, that is the major part, which is about 75% or 70% of the gland is composed of this anterior lobe, pars distalis. Then there is intermediate lobe, which is this one, pars intermedia. And the third is the tuberal lobe or pars tuberalis, which is that part which surrounds the infundibulum. The neural component of the stalk is surrounded by this extension of the adenohypophysis, and that is called pars tuberalis. These are the three parts. One is the anterior lobe. This is the anterior lobe. This is pars intermediate. That is intermediate lobe. And then this is the pars tuberalis around the pituitary stalk. These are three components of the pituitary gland. Pars distalis, that is the anterior pituitary, anterior lobe. It is also called anterior lobe. That is about 75% of the gland. 70 to 75% of the gland is composed of this important segment. It is highly vascular gland. About 50% of the cells are chromophobes. That is, they do not stain, neither they stain with hematoxylin nor with eosin, and they are chromophobes. 35% are acidophils, and they secrete growth hormone and prolactin, and 15% are basophils, which are concerned with the secretion of ACTH, TSH, FSH, and luteinizing hormone. These are the other hormones, glycoproteins, which are released from the basophil cells. So overall, this is the cell. This is the region which constitutes 75% of the gland. It's very vascular. 50% of chromophobes, 35% acidophils which secrete growth hormone and prolactin, and 15% are basophils which secrete adrenocorticotropic hormone, thyroid stimulating hormone, follicle stimulating hormone, and luteinizing hormone. Pars tuberalis is around this. The cells of this, the function of the cells in this region is not known. Pars intermedia, these are the cells which contain colloid-filled vesicles, and they secrete melanocyte-stimulating hormone, and it also contains endorphins. So these are the characteristic features of pars intermedia. So three parts of the adenohypophysis have different functions. The function of the pars tuberalis is not known, and this is concerned with secretion of the important hormones which are controlling the thyroid gland, pituitary gland, and gonads, suprarenal glands. All these are controlled by the secretions, trophic secretions, which are formed and secreted by the anterior pituitary. Pars intermedia is concerned with secretion of melanocyte-stimulating hormones. Neurohypophysis, this is the posterior part, and it consists of, again, three regions. The larger one is the posterior lobe, which is called pars posterior or neural lobe. Pars posterior, posterior lobe, or neural lobe. Then there is stalk, infundibular stem, and then there is median eminence. This region is the median eminence. Right? These are the three parts of the neurohypophysis. Infundibular stem, this one, it contains neural connections. Neural connections means it is composed of fibers, axons of the cells, the cell bodies of which are present in the paraventricular and preoptic nuclei. So these are the mainly the fibers or axonal fibers of these hypothalamic nuclei, that is paraventricular and supraoptic nuclei. Their axons are present in the infundibular stem. They are neural connections of the posterior lobe with the hypothalamus. Pars nervosa, this pars nervosa, it contains about 100,000 unmyelinated nerve fibers. Unmyelinated nerve fibers, you see that these nerve fibers are coming from these nuclei. The cell bodies of these nerves are present over here. This is the paraventricular nucleus, and this is the supraoptic nucleus. And their axons are coming over there, and cell bodies are present in the suprachiasmatic and paraventricular nuclei. And they secrete antidiuretic hormone and oxytocin here, respectively. These are the hormones which are released, which are manufactured in the cell bodies over here. Then they are released, and they are conveyed via the cell fibers at a rate of about 3 millimeters per day. They go into the posterior pituitary, right? Posterior pituitary itself contains the cell bodies of the pituicytes. Pituicytes are astrocyte-like cells which are present in the posterior pituitary. So here you can see the paraventricular hypothalamic nucleus, and which releases oxytocin, and this is for the antidiuretic hormone. The supraoptic hypothalamic nucleus, and they contain the hypothalamo-pituitary tract is formed, right? And then the secretions are poured into the pars nervosa, from where they are distributed to the bloodstream. And this is the anterior pituitary. And these cells, infundibular nucleus cells, these cells which are present here, they are concerned with releasing or secreting the releasing hormones which go to the anterior pituitary to activate or inhibit the secretions of the cells in the anterior pituitary, right? So these are the hypothalamic cells which control the secretions of the anterior pituitary. These are the cells which pour their secretions into the posterior pituitary. So this was all about the adenohypophysis, its morphology, the main types of the cells, its location, its relations, and the activities of the, we'll be discussing about the activity shortly. Activities you can predict by the hormones which are released from these areas. For example, from the posterior side, oxytocin, which is released into the circulation, that oxytocin is for the contraction of the uterus during parturition, or for the, it is also useful for during lactation, right? The supraoptic hypothalamic nucleus, which releases antidiuretic hormone, that is antidiuretic means it inhibits diuresis, and when it is stimulated, it retains water and salt, right? Similarly, these are the releasing hormones which either cause the release or the inhibition of the hormones from the anterior pituitary. Growth hormone is for the growth. Then again, prolactin is concerned with the milk ejaculation, and similarly, thyrotropic hormone TSH is for the stimulation of thyroid gland. Adrenohydrotropic hormone, ACTH, adrenocorticotropic hormone, is for action on the adrenal glands, right? And FSH and LH, they act on the gonads. And these are different functions of the glands. Now, blood supply of hypophysis cerebri is very typical, particularly of the anterior pituitary. There are more than one superior hypophyseal arteries and usually one inferior hypophyseal artery, which both arise from the internal carotid artery. Superior hypophyseal artery, here is shown one, maybe more than one, which goes and then it breaks up into capillary networks, right? And the hormones which are coming from the hypothalamus, releasing factors and inhibitory factors, they are poured over there into these capillaries. These capillaries then reunite to form the venules, and these venules in the anterior pituitary again break up into the capillaries, and these releasing hormones are poured over there where they act on the cells of the anterior pituitary. Then again these reunite to form the capillaries which are poured into the collecting veins or into the cavernous sinuses usually, right? So what is the specific thing about this sort of arterial supply or venous drainage? This is a typical example of a portal system, venous portal system. Here it is called hypophyseal portal system. Hypophyseal portal system. Portal system is actually a system of vessels in which between the arterial end and the venous end, two sets of capillaries are present. Normally, there is one set of capillaries. Blood coming from the heart goes to the periphery or to its desired location, and then breaks up into the capillaries. Then capillaries reunite to form the venules, and then veins, they go into the heart, right? This is the normal systemic type of circulation. But at some place in the body, there are two sets of capillaries, right? First, the blood comes from the heart via its branches, as for example here, by the branch of the internal carotid artery, the blood goes into the pituitary stalk, and then it breaks up. The vessel breaks up into capillaries, right? Then these capillaries reunite to form the veins, and the veins then again break up into the sinusoidal capillaries. And again, then they unite to form the veins. So there are two sets of capillaries are present. One is over here, the other one is over here. So this is called hypophyseal portal system. This is specific for the hypophysis cerebri. And another very important system is present, which is called portal system, portal vein. You might have heard about the portal hepatic vein. So that is portal hepatic system, right? So posterior pituitary, on the other hand, it receives superior hypophyseal artery and infundibulary artery and inferior hypophyseal artery. These are separate branches, and they normally the artery is breaking up into veins, and veins are again venules are again united to form the veins. It's a simple sort of circulation. Released from here, or the hormones here, and then it is released into circulation, and via these vessels it goes into the systemic veins, right? So anterior pituitary has got the hypophyseal portal system. Posterior pituitary does not have this side of posterior portal system. So here you can see the portal system where two sets of capillaries are present, and here there is no portal system in the posterior pituitary. Here you see suprahypophyseal artery is dividing into capillary network. They are again unite to form the larger veins, which are again breaking up over here, and then they are releasing into the cavernous sinus. The blood will go into the cavernous sinuses. So this is all about the pituitary gland. Any question is welcome. Anything which is not clear, you can ask me. Sakina has asked, can you please review the chromophils and chromophobes? Right, chromophils and chromophobes. Chromophobes are the cells which do not, when we stain the gland with hematoxylin and eosin, so chromophobes are those which hate the dyes. That is, they don't take any stain. Chromophils are the cells which are mainly responsible for the secretion of the these chromophobes are about 50% of the cells over there, and chromophils, they are 35% are acidophils, that is they are stained with the eosinophil, and 15% are basophils, which are stained with hematoxylin. Now, chromophils, they are concerned with, acidophils are concerned with the release of the, as I told you before, for the growth hormone and prolactin, and the basophils are for the release of the other hormones, that is follicle stimulating hormone, luteinizing hormone, adrenocorticotropic hormone, and thyrotropic hormone. They are released by the acidophils, basophils, sorry. So this is the difference. Difference is in the staining. I hope it would be clear. Any other question?